Hello, this is Tommy. Welcome back to Chatnomics. In today's video, I'm going to show you step by step how to download TCGA bulk RNA sequencing data and make a PCA plot. And also in the end, I will show you how to make a heat map based on the downloaded RNAseq data. So what is the TCGA? So TCGA is short for the Cancer Genome Atlas. It actually characterized over 20,000 primary cancer and matched normal samples spanning 33 different cancer types. And actually it includes different modalities of data types. For example, RNA sequencing, mutation data, copy number data, DNA methylation data, and also even proteomics data. So it's a very comprehensive data set, and it's actually freely available for you to download and explore. And you will read actually many papers using TCGA data to generate a new hypothesis and answer new questions. So it's actually very good data sets for you to practice your bioinformatics skills. So in today's tutorial, I will show you how to download the data using an R package called TCGAbiolinks. And there are many ways to get the TCGA data. You can go to GDC, download them directly, but the TCGAbiolinks package makes it very easy to download the data and return it as a summarized experiment object in R with associated metadata information. Okay, so in this tutorial, I will download the lung adenocarcinoma bulk RNA sequencing and also the lung squamous carcinoma and RNA sequencing data and combine them together and make a PCA plot. Okay, make sure you stick to the end. I will show you even how to make a heat map to separate those two cancer types. Although they're both lung cancer, but they are of different subtypes. Okay, so if you don't have those libraries installed, you have to go to just Google and it will tell you how to download it. It also has very comprehensive tutorials how to use the package. Okay, how to get clinical data, how to get the mutation data, for example. Okay, I already have them installed, so I will just load those libraries. Okay, and so this is the function you're going to use called GDC query, and then specify the project name, TCGA LUAD, which is short for lung adenocarcinoma. The data category will be transcriptome profiling. Data type will be gene expression quantification, and the workflow type will be the star. We'll get the raw counts. Okay. So usually this takes a little time because it will access the GDC database. And then after you finish the step, and then you use GDC download to download all the raw counts. And the data is actually kind of big. It actually take a little bit of time to do that. So I already finished this. So I saved this actually. For example, after you download data, use this GDC prepare function to return a summarized experiment object. And then I saved it as an RDS. I already saved it, so I will just read it back. Use the read RDS file. Okay. You see, actually, it's kind of big. Let's see. Okay, it's done. And now you see, like we're using a 2 GB actual memory here. And so if you look at this object, it will tell you this is a arranged summarized experiment object. So this is an extension of the summarized experiment object, adding the gene informations like where is the start, chromosome start and end for each of the genes it mapped to. So there are many actual metadata for each of the sample. Okay, we can use the core data function, and if you do this, it will show you just all the metadata information. It's a lot. So we can look at the column names, and you have actually 89 column names. And if you just look at a couple of them, it will tell you some of the metadata information, for example, the purity from the paper that published these TCGA data sets. And then, for example, it will tell you also the DNA methylation signature, for example. Okay, so we can take a look at the CPG, for example, same methylation signature. It will tell you whether it's high, median, or low, and how many of them are of each category here. So I'm using the janitor this package and use this is just a different table function, and you can use that directly within the data frame. I always use that. Okay, let's save the raw counts into a new matrix. Okay, so if we just use the ASCII function, and this is the summarize the ranged summarized experiment object. So if you use assay function, and then it will give you a data matrix. So if you look at the first 5 rows and the first 5 columns, the rows are genes, columns are samples, and you notice that this is on sample ID. Also, it has this version number actually in the end. So we need to actually remove this in order to convert this on sample ID to gene symbols. I will show you how to do that as well. So if you look at the dimension of this matrix, it has 60,000 some rows, essential genes, and you have 600 columns, 600 suppose. Okay, so we will do the same thing for the function GDC query, but instead of TCGA LUAD, now we're using LUSC, and the rest are the same. And download the data, the raw counts, and use the GDC prepare to get the summarized experiment object. I saved it already. Now I'm just reading it back. It takes a couple of seconds here. Okay, it's done. And see the memory actually increased to 3.45, so it's roughly probably 1 gigabyte for that object. And if we look at this, now we look at the metadata, and actually we can look at the expression subtype from the paper. For example, it will tell you basal, classical, primitive, and secretory. So those are the transcriptional subtypes from the TCGA paper. And of course, there are also a big number, actually, there are NAs. I guess it's like when they publish, they only have those data available. Okay. But of course, you can actually impute those unknowns to each of the subtypes. Okay, anyway, so we can also save that as a matrix, a USC matrix, use the assay function again. Now we need to actually convert that ensemble ID to gene symbol. So let's do that. We should actually load this organism actually database. So if you take a look at this, it actually contains all the information for you to map the different gene IDs. Okay, let's do that. So this is the matrix we have. If you do that, that's all the gene names, and we use the table function in frame. So this will actually give you a data frame or a table actually. So we convert that single vector to a table or table here, and we will mutate this column and remove all the digits in the end. So let's see. So we use the stir column as the first argument, and then replace this dot and then all those numbers. There's empty string here. So if we take a look at this and look at so this is what it looks like here. Now we are ready to use this column to map the ensemble ID to gene symbols. So I will use the BITR function from the cluster profile package, and this is the input, the ensemble ID, and from ensemble to symbol, and the organism database will be the human doc.hs.eg.db that we loaded here. And actually there are actually duplicate gene symbols for different ensemble IDs. So if you take a look here, use the getDupes function, And after we map, so let's take a look at here first. So always build the pipe actually step by step, and so you understand what's the output. And you see actually this output. So we have, so actually what we can do, because it print out all the 6 rows here. So it will give you this actually, the ensemble ID, and then also the symbol. Okay, so we can use this get dupe function and the symbol, this column, and see which one are duplicated. And you will see here, for example, this symbol, it appears like 16 times, but they are mapping to different ensemble IDs. Okay. So what we will do actually, we will just use the distinct function and just take one of them, just make it simple. And so let's do that. And now we have like a one-to-one mapping for the ensemble ID and the symbol. So we actually want to left join this data frame here, left join here. So let's see what we have. Now we have that TCGA LUAD map. So this is what we have here. The ensemble ID without the dot, the last several digits here, the symbol. And now we can use this to subset the expression matrix and replace the row names to gene symbol, right? Remember, this is the old data matrix. Oops. 125, right? And now we use, this is the gene mapping data frame we have here. So we use this column, value column, to subset that data matrix. And because now we have the gene symbol that is mapped to this, so we can just change the row names to this column. Okay, so let's do that. And if you look at the first 5 rows and 5 columns, now the rows become the gene symbol, and columns are still the same of the samples. So we do the same thing for both LUSC and LUAD. Okay. So now double check the dimensions of those 2 matrices, and they are the same. Okay. Now we end up with 37,000 genes, and then we have 500 some samples for the LUSC and 600 for LUAD. Okay. Now we can combine those 2 data sets just by concatenating them. So let's make sure all the row names are the same, and it's true. So now we have 2 actually data matrix with the same row names but different columns. So you can just use the C-bind to bind it to a bigger data frame, the bigger matrix. So we just use C-bind and assign it to the TCGA_long matrix. Okay, it's a new variable. And at the same time, we can actually create a metadata. So we can say, okay, what's the cancer type? The cancer type will be LUSC, and n column of LUSC. This will be 553. So it should replicate or repeat LUSC for 553 times, and the same thing for LUAD, and then for 600 times, I believe, here. Yes. So if we do that, so this is a new dimension, gene by sample, and this is just tell you, okay, those are which sample are from which data sets, from which cancer type, right? LUAD or LUSC. Okay, now I will show you how to make a PCA plot. Okay, so you use ggplot2 load them, and then use the G45 library as well. Okay, in this case, we have a new matrix. Now we will actually just select the most variable genes across all the samples in those two data sets in the combined matrix. So what we can do, you can use the row vars function to calculate the variance of each row, and we used order function to order from high variance to low variance, and let's just take the first 1,000 most variable genes. So this is just an index. So if you take it here, it's just the index of the top 1,000 most variable genes. Now we can use that matrix to subset the original matrix, which has like over 30,000 rows or genes. And we do this, and let's take a look. That's the dimension here. So as expected, we have 1,000 genes, rows, and columns are the total number of samples for those two different cancer types. Now we can use the PR comp function. This is the base R function to calculate the principal components. And to do this, actually, we need to transpose the matrix first. So this is actually a gene by sample. So usually people, I think PR comp, it works on both ways, but now it's better just to transpose, because you will get the right actually components for the samples, for example. So we need to actually transpose it. So if you actually don't want to learn a little bit more, I think I have a link here for the PCA analysis, like here. Okay. So by the way, I will have this script in the description of the video so you can follow along. And so we need to transpose it first to sample, rows are samples, columns are genes, and then do a PR comp, principal component analysis. And this is actually, so if you look at this, it's a PCA project object. So it has different components here, standard deviation here, rotation, sentence, scale, and also the X. So this actually contains the principal components for each sample here. So if you look at the first 5 rows and first 5, so you have PC 1 to 5 here and the first 5 samples here. So again, make sure you go and read this post if you want to understand more about the PCA. Okay, now we have that. So you can, in ggFortify, you can just, after you load it, you can use the autoplot function and give the PCA results. And this is the metadata. So remember, we have this metadata to indicate that those samples are either LUSC or LUAD. And then we, use color equals to cancer type. So, and then we manually set, red, blue, and the red for those two cancer types. If we do that, and you see here, now we have the PCA plot, and PC1 here, PC2 here. And what you see here is that somehow those two cancer types, they are separated in the second principal component, although there are some actually LUSC here that is mixed with LUAD. So what's going on here? So because we just use the raw counts and then we find the most variable genes and do a PCA. So we are actually missing one step, which is normalize to the sequencing depth or the total number of counts for each sample. Okay, we need to normalize that because different samples, they are sequenced to different depths. Okay, so actually the first principal components here is correlated with sequencing depths. I will show you how. So again, let's look at the principal components here, and the sequencing depth here will be just the column sum of that TCGA matrix. So this give you the total number of reads for each column. So we save that as a new variable here. Now we can calculate these first principal components, the correlation between that. So if you look at here, so essentially this column here, because for each sample you have one value for first principal component, and they also have the sequencing depth for each of the sample. Now you can calculate the correlation. And look at the absolute value here is like 0.979. So it's highly correlated with the sequencing depths, right? But if you look at the second actually principal components essentially here, and then if you look at the correlation, now it's actually not well correlated, okay? So we can actually plot that in the PCA as well. So we can add a new actually sequencing depths to that metadata data frame. So now, if we do this, now you have two columns here. One is for each row, actually for each sample, you know the cancer type, and also have the sequencing depths. So now you can actually do the PCA plot, but now instead you're coloring the sequencing depths, and you can see here the sequencing depth from low to high, right, from left to right in the first principal components here. Okay, so now let's actually normalize it to counts per million. It's a very simple normalization methods, and see how it looks like after PCA. Okay, so we use the EdgeR package. It has a CPM or counts per million function here. Give the matrix, log2 transform it, and use 1 as a pseudo count. So log2 actually CPM plus 1. Okay, let's do that. So actually you can just calculate even just manually by yourself. Counts per million is just essentially just get the column sum of each column, and what it actually did that is the sequencing depths, and for each value divide that number and times a million. That's counts per million. So okay, and then we'll do the same thing. Just choose the top 1,000 most variable genes for that counts per million matrix, and subset that, and let's do a PCA again. Use the PRComp function, and now we can actually plot the PCA plot again. Okay, again, use the color to indicate which cancer type it is. Okay. Now we see, okay, the lung adenocarcinoma and the squamous carcinoma, they are kind of separate in the first principal components here, okay, which makes sense. However, it's actually very interesting to see those actually LUSCs, they kind of get intermingled into the LUAD. Okay, so anyway, that's actually some further homework you can do. So what's going on for those actually LUSC samples? Now let's make a heat map. Okay, using the genes that most variable in this whole data sets. So I always use complex heat map to make heat maps and read the tutorials from the bioconductor package. And it's very comprehensive, and you can make any heat map that you want to make using complex heat map. So it definitely worth your time to invest and then learn how to use this package. Okay. So I'm going to actually have this top annotation. So I'm using the data frame and TCGA long metadata, and the cancer type is USC with red, LUAD will be blue. So you have to read that documentation to know how to use this heat map annotation function. Now I'm going to actually just scale the CPM, the subsetted CPM matrix, the counts per million matrix, and scale it, and then send it to heatmap function here. And this top annotation is this one that we just defined. I also did a k-means clustering for the rows or for the genes here. Okay, let's see how it looks like. I need to actually run this line first, and let's see. Okay, it's down here. Okay, so we have the LUAD and the USC kind of nicely separated into two different groups here, right? And also there's some actually LUAD here that's kind of mixed with the USC here. So, and also vice versa, you have those LUSCs that can intermingle with the LUAD. Okay, but again, this tutorial just show you how you can practically download the TCGA data and do a PCA analysis, and do a heatmap visualization. And in the future tutorial, I can show you how to do differential gene expression analysis using TCGA data. Make sure you subscribe to this video, to this channel, Chatomics, and keep coming back and see the new tutorials that I will be preparing for you. Okay, that's it for today, and happy learning. I will see you next time.